Testing, testing. Oh, I need to share screen. Yep. Okay, great. Oh, well. I think I've got two things open at the same time. Let's close this one. And we will go share. All right. I think we're good. Yep. Yep. I think everything's good. Yeah. Hey, sorry. It's running around a little bit crazy today. I forgot that today's not Thanksgiving, Halloween. And so, yeah. I have children, and so hence it's a big thing tonight. Yeah. Well, presumably if they want people to give them candy. I mean, I dressed up until I was like 20, but then around that time I used to go to like McDonald's to trick or treat. Like a group of us, like college kids from Stanford, we all just walked into downtown Palo Alto. We walk into McDonald's, we're like trick or treat, and the manager's like, well, I guess you can have anything you want off the dollar menu. Done. I mean, they just wanted to get rid of a bunch of like so. Sure. Sounds good.
Thanks for joining the seminar today. We're very happy to have Mike Bloom, our seminar speaker. Mike is a professor in biostatistics of the Public Health Science Division at Fred Hutch Cancer Center, and also affiliate professor in our department. And one of the major research areas focus on developing and applying statistical methods for addressing arising in the omics science, including genomics, epigenomics, proteomics, and metabolomics, but usually not economics. Our recent focus is on the recent focus on Mike's group is on the human microbiome, and through this line of research, there are many tools developed which have been integrated into components of many analytical pipelines. And in a separate line of research, Mike also participated in SWAP, which is a cancer consortium and a member of the SWAP Statistical Center. And without further ado, let's welcome Mike.
Okay. Thank you very much, Ting, for the very nice introduction. I can't remember if I wrote that or if, okay. Actually, I didn't write it. Somebody else wrote it for me somewhere else. And then everywhere asks you for these intro slides, and you never want to write it yourself because it sounds stupid. And so I just send them whatever. This one, I think Toronto wrote for me at some point. All right, anyway, so today I'm going to talk about an area in which I know almost nothing. That is like the best type of talk that you can give when you don't know anything. And then you just hope the audience knows even less than you. Like, that was like most of my job talks, to be fair, but it's like, and I got a job, so I guess it kind of worked out. But we're going to do something along the same lines today. So I'm going to talk about the analysis of spatial proteomics, and we're going to talk about the mysteries of space and time. Woo! Yeah. Okay. And this is my slide advancer is not working properly. We will see if we can fix that. There we go. So here's an overview of the presentation. I'm going to talk a little bit about motivation, why I'm doing this. I'm going to talk about analysis of spatial proteomics data. In particular, I'll talk primarily about a new spatial omnibus test that we've developed. I'm going to skip over this, probably because I'm not going to have time. I came up with these slides when I was really, really ambitious. And then I was like, I think 200 slides is like way too much. But anyway, then I'm going to talk about a new area, which is harnessing this area of research called topological data analysis to analyze spatial proteomics. Topological data analysis is commonly done in mathematics, applied math, as well as electrical engineering and a number of other areas, but we don't see it very much in statistics or biostatistics. So I'm excited to draw some connections between the things that are done there and some of the stuff that we do more commonly in statistics. All right, so here's the motivating study over here. It's called SWOG study S1616. That means nothing to you. All right. SWOG is a group that I work with. We coordinate clinical trials that take place around the country. Right. So these are big, important clinical trials. So 1616 is a study of metastatic, which is late stage melanoma. And so this is a melanoma. This is probably a less hideous looking melanoma than you will see. You will see melanomas that are taking up people's entire faces. When you go to these medical conferences, I think they must compete to see what sort of nasty pictures they can put up there. Because it's like, yeah. And then they're like, hey, have breakfast with this seminar. It's like, yeah, no, thank you. Anyway, so back in the day, like probably around the time many of you were born, metastatic melanoma was horrifying. If you had stage 4 melanoma, it was basically a death sentence. All right. You were counting your time in months if you were lucky. All right. There were treatments. People had come up with these really, really toxic things, dacarbazine, interleukin-2, interferon alpha. All right. These are really, really toxic chemotherapeutic agents, and they didn't really work. People still do them because, well, you have nothing else. Like you talk with melanoma docs, they were like, yeah, it was a sad time. You'd see a patient and you know that they're not going to be here in a year. All right. Then around 2011 to 2013, all right, suddenly an entire new class of drugs appears. This is called immunotherapy. All right, three dudes won the Nobel Prize for this. Why? Because it actually works. It works very well in melanoma, in lung cancer, in bladder cancer, colorectal cancer, many, many different types of cancers. And the ultimate idea behind this is you harness your own immune system in order to attack the cancers over here. And so since then, in the last decade or so, there's been tremendous progress and people come up with more and more drugs along these lines. Interest is starting to wane, mostly because the drug company patents are running out. And unfortunately, much of research in clinical trials is partially driven by drug company interest, which is unfortunate. But and so, oops, that was just to say people used to die back then. I forgot that. All right, so here is a particular type of immunotherapy. It's called anti-PD-1, and so this is one that is quite popular. It works pretty well, all right? And then there's another one where you combine it, anti-PD-1, with anti-CTLA-4. Don't need to know the details, just one targets one thing, one targets two things, all right? 1, 2, those are numbers, all right? That's all you need to know. Right? And so these are options for patients with advanced melanoma. All right, so anti-PD-1, this is number 1, all right? It's great, all right? You went from response rates of approximately 0 to approximately 40%. So 40% of patients with late-stage melanoma are now responding to treatment. Yay! If you combine that with anti-CTLA-4, so now you've got 2 drugs, the response rate is about 60. All right, and you're like, yay, even better. 60 is better than 40, all right, especially when you're talking about response rates. So everybody's very happy, right? No. Here's the dilemma, all right? The combination therapy does have a higher response rate than monotherapy. So this guy, better than that guy in terms of response rates, all right? If you are somebody with cancer, you're like, yeah, I would love to get this, right? Because more people respond. But the problem is the combination also has much higher toxicity. So you're taking, what happens with these immunotherapies is you take the brakes off of your immune system. Intuitively, that's great when you've got cancer because then your immune system goes bonkers and then goes and kills all the cancer. But the problem is also that your immune system is going bonkers. And so it has huge numbers of immunologic side effects as well. Like, you know, sometimes your immune system will start targeting you. I mean, this is what we see when we talk about lupus and other sort of autoimmune diseases. All right. And, you know, it sounds great, but the toxicity is really a problem. So the idea here is that if you have this combination therapy, it may be too toxic for routine use, in particular because monotherapy already works pretty well. I mean, 60 is better than 40, 50% better, but it's like, are you really willing to risk? Sometimes these are older patients, people who are already pretty frail. Are you really willing to give them these really toxic things? Well, so the idea behind 1616 was the following. Let's try something where we treat with ipilimumab, so that's CTLA-4, after somebody has primary resistance to nivolumab, which is the PD-1. All right. So very, very simple. All right. What we do is we start you off with just one drug. All right. If you respond, great. If you progress, which means your cancer gets bigger, gets worse, then what we're going to do is we're going to give you the combination, and hopefully that'll reverse the resistance to just the cancer's resistance to this drug, right? So 1616 was a phase 2 randomized trial to compare combination therapy versus CTLA blockade in patients with primary resistance. That's just the title. Okay. This thing, you guys probably don't care. It's pink mushy stuff, biology. It matters to the clinicians I talked with, but you guys don't really need to know about this. So here's the trial schema. We take patients with advanced melanoma that has already progressed. So their cancer has already gotten worse when they only took one drug, all right? Then we randomize them 3-to-1, three patients to the combination, 1 to just a single agent drug over here. And then our endpoint over here is progression-free survival. We collected all sorts of strange and wonderful things for correlative studies and all sorts of fun things there. Okay, and these are the clinical trial results. So this is progression-free survival, time until your cancer gets worse. Looks pretty bad, right? These are your Kaplan-Meier plots. This is the percentage alive without progression. And you can see within 6 months, the vast majority of patients have progressed. But patients who are on this combination arm are still doing better than patients on just a single agent arm over here. In fact, if you look at this, this is about 20% over here. And so if you think back, 40% were responding in the front line. And then if you can get 20% of the subsequent ones, this is actually not terrible over here. All right. And the overall analysis, we designed this for a one-sided 0.05, two-sided 0.1. And the p-value over here is 0.037. Less than 0.05, which is what we designed for. So yay, everybody was happy. And in fact, the field was very happy. And this appeared last year in Nature Medicine. Okay, all right, which I don't really care, but the biologists seem to care a lot about that. But yeah, anyway, I was just happy. I was like, okay, this thing is off my desk. I don't have to think about it ever again. Wrong, I had to think about it a lot more. Attached to these trials are what we call translational medicine, which are correlative studies. And the idea behind this is to understand what is really going on mechanistically inside of the body. Why is it that some patients respond, some patients do not over here? And so we collected baseline biopsies from all of these patients, and then we collected tons of different types of data on there. We have bulk RNA sequencing, exome sequencing, TCR sequencing. We have spatial transcriptomics on hundreds of samples over here. And we have what is called multiplex immunohistochemical staining and MIBI over here. And that's the data type that I'm going to focus on today, all right? Because this is what we call spatial proteomics over here. So the first project I'm going to talk about is how to analyze some of this. And I'm going to talk about a method called SPOT, or this, where we're looking at the spatial distribution of cells within a particular tumor over here. So what is spatial proteomics data? Well, here it's space. So here's where space and time come into play. Space because it's spatial, time because we're talking about the tumor immune microenvironment over here, all right? So the space-time continuum over here, the question is, does time composition, the tumor microenvironment, so that's what's going on around the tumor, inform the outcomes over here? And so spatial proteomics, what happens is you generate antibodies for specific proteins or markers. And then these guys have little fluorophores that are attached to there. You basically wash it across these samples, and then you image them over here. And so if these antibodies for particular proteins stick onto there, then that means the protein is there and it lights up. All right. And so what you end up with is an image for each patient or each sample. And you get a bunch of different markers, and you can detect each of these guys over here. All right, and so after image processing, they're just standard software for this. The next thing that people typically do is spatial analysis. So here's an example. This is zoomed in. So we had a whole bunch of different markers that we looked at, and then based on our knowledge of what cell types express which types of markers, we can determine whether we have over here a CD8 T cell, over here, this guy over here, a tumor cell, all right, or you have a dividing CD8 T cell over here, and so on and so forth. We can actually get pretty granular over here. This is actually very different than spatial transcriptomics, which you've probably seen a lot more work on. The main thing is we actually know what we're looking at here because we chose these markers a priori. So we know exactly what these different types of cells are over here. When you do spatial transcriptomics, you're just looking at all of this stuff mushed together, and then you hope your t-SNE or UMAP is dividing out something that may or may not make sense. If you talk with Lior Pachter, he thinks nothing makes sense. If you talk with other people, then they still believe in it. I don't know. I don't analyze those data because I think it looks like crap. The other difference here, it tends to be lower dimensional, and we have tons and tons of samples. If you look at the stuff that a lot of people in spatial transcriptomics are publishing, basically they take one sample and analyze it to death. That's just where the field is starting. People are starting to do different things. The other thing, we actually have subcellular resolution of where these proteins are, which can give us other things, interesting things going on. So it's just a detour, just saying this is different than spatial transcriptomics. So don't sort of combine these two concepts together at the same time. All right, here's an example of what our data actually look like. For the most part, what I'm going to talk about initially is just where the cells are located. And that is mostly what people do. So here's an image. It's just a table. All right, if you have an image 1, all right, image 2, so on and so forth. All right, and then you have cell 1, 2, 3, 4, up to however many cells are on this slide. And then it tells you what is this type of cell. All right, it gives you the X and Y coordinates where they are on this slide. All right. The tissue type over here, is it in the tumor? Is it in the stroma? The age of the patient. So these are cell level things over here. All right, this is image level. All right, and this is basically patient level over here. So you have age, you have their survival status, and their cancer stage over here. So the idea here is, between spatial analysis, is that we're interested in understanding how the spatial distribution or arrangements of cells, their XY locations, as well as the types of cells that they are, and how that is related to patient outcomes over here. Okay? So what do people do with this? The most common strategy to do this is, well, you summarize the spatial distribution of the cells using a simple spatial summary statistic. Okay? It's just a spatial summary statistic. This is different than brain imaging. Everybody's brain sort of looks the same. Everybody's tumor and the biopsies that you get out of there are totally different. There's no registration. There's no matching up any of this stuff over here. So we just use some sorts of summary statistics over here. Typically, they describe the degree of clustering of particular types of cells, the degree of repulsion, or how relative they are to complete spatial randomness over here. Are they just totally spread out in a totally random way, or is there some clustering over here? Then you take these spatial summaries, you have one for each sample, and you just treat it as a covariate in whatever linear regression, logistic regression, Cox regression, or whatever fancier regression model you want to do. Nothing terribly fancy going on. What are the types of, how do we typically do this? All right. So if you only have a single cell type, so let's pretend we're only looking at CD8 cells, then what you do is, most commonly, is you assess whether the cells are clustered more than expected as compared to complete spatial randomness. And standard metrics include Ripley's K, Bissog's L, Geddes or G, Shoge's J, okay, Herdes M. There are more letters than you can come up with over here. For the record, Julian Bissog was a faculty member here. I never met him. He apparently, but if you look at his memorial, the most pleasant thing anybody said about him was that he was a deeply unpleasant individual. All right. And he apparently was a super alcoholic and died of alcoholism-related issues. So anyway, yay our stat department. Okay, so how do these typically work? Well, what you do is typically you take this segmented image, all right? We also have markers for the cell membranes. And then what happens is you put a dot in the middle of each cell, and then you basically draw a radius around there. And then you can count up, for example, the number of cells of a particular type that are within that radius. And then you do this for every pair of cells, and then you can just count them up and compare that to what you would expect under complete spatial randomness. So that's Ripley's K. Biosog's L, he divided it by pi and square rooted it. All right. And he got another paper out of it. So yay him. All right. Then there's other weirder things that people come up with. There are all sorts of different metrics that people come up with over here. People seem to use them. They seem to like them. Whether or not you like Ripley or Biosog, I don't know. I don't care. It's whatever the biologist wants over here. Okay. So take a point. You draw a radius around it, and you move from there. What's the issue over here? So here's an example of what's going on. This is one where there's a high Ripley's K, so that you think that there's a high degree of clustering of this immune cells. So the red cells are immune cells. The other ones are just background tumor over here. And you can see that they are sort of clustered together, versus here, it's much more similar to complete spatial randomness overall. And so if you take a look at the radius, all right, and then you can look at the Ripley's K as a function of the radius over here. So the blue line is complete spatial randomness. This red line over here is the Ripley's K that's observed in this particular image across the range of potential radii that you could use. You can do the same thing in this one, and you can see that it's much closer to complete spatial randomness over here. So intuitively, very, very straightforward. Are you seeing some sorts of weird clustering, or is it everything just sort of mixed up randomly over there? What's the challenge with this? Well, it ends up, we can extend this. All right, that's just with one cell type. With two cell types, typically what we're doing is we're assessing whether cells co-localize. The idea is if I always have a CD8 cell and it's always sitting next to a dendritic cell, then that suggests that there's an interaction between these two different cell types. All right, so it's very, very straightforward. If you see a statistician and the statistician's always standing next to an accountant, then you think, hey, they may be, the statisticians and accountants might interact particularly much over here in a room, versus if they're all just scattered together in this giant room full of random people. So bivariate versions of these statistics also can be developed over here. And so here's, Ripley's K, a bivariate version, and Biosog's L, Gedesor G, and so on and so forth in these cases. Critical to all of these is a radius. So each of these, you have a radius over here. And so one of the challenges is in how do you actually choose the radius at which you want to do your analysis. Okay, this is a problem that a number of individuals have undertaken, in part because the choice of radius actually heavily influences the analysis, and you will get different results depending on which radius you choose. The best or most biologically relevant radius is typically not known a priori, and it's contextual. All right, these cells secrete things. Other cells secrete things too, and then you don't know how they're really interacting, sort of biochemically in this case, a priori, and it depends on the types of cells. So it's really, really messy to do this, and there's no clear guidelines for selection across applications, which is why we proposed a very, very simple approach called SPOT. This was the first project that we worked on this, and it was mostly a learning exercise. But the idea behind this is very, very trivial over here. Right? It takes about a week to do this paper. So here's the procedure. What you do is you select a broad range of candidate radii. You calculate spatial summaries at each radius. All right. Then what you do is you assess the association between the summary and outcome at each radius. You get a p-value. All right. Then, so for each radius, you now have the p-value for the association between the summary statistic and outcome. Then what we do is we just aggregate the p-values by something that we call a truncated Cauchy combination test. All right, nothing fancy to this at all. All right. So the result over here is we end up with a single omnibus p-value for the association. And because we've looked across all of these radii, we actually have a better understanding of a so-called best radius, which has actually been an interesting biological byproduct of this project. So the Cauchy combination test, this is a very well-established approach now. It was from 2019. given a set of correlated p-values, p1 through pk, in this case corresponding to each radius, the idea is that you want to get a single p-value. Now, if these guys were independent, then you can just use a Fisher's combination, whatever standard, easy, old, like 1930s thing you want to do. All right? But the problem is that they are correlated. People have come up with ways of combining correlated p-values. None of them will really work very well until there's this Cauchy combination test. And there's also a harmonic mean p-value that came out around the same time that both seem reasonable. But we did this just because it's easy and because it has the Cauchy. Everybody hates the Cauchy. Well, now everybody loves the Cauchy. All right. It's always the pathologic example in your introductory probability class. And now people are like, wow, this is awesome. So the idea here is you take these p-values and you transform them into a standard Cauchy. All right, p-value back to Cauchy, that's just your standard intro probability, whatever integral transform thing. All right, and so you can just do that, right? So you get a test statistic. This is, you take your p-values and you back transform it to a Cauchy, standard Cauchy distribution. Then what we do is we basically take a weighted average of these Cauchys over here. All right, and it ends up that these weighted average of Cauchys is still Cauchy over here, right? Under the null. So P1 through PK are uniform, and then under the null, these guys over here go back to a standard Cauchy distribution over here, okay? And so the issue here is that the challenge here is typically that classical p-value combination approaches require some degree of independence among the p-values. You can do permutation testing. It generally works, but it introduces other problems and it's ridiculously slow. And then there are approximations that people have used, which tend to focus primarily on means and variances, which don't work very well in the tails, which is what I really care about. I don't care about a p-value of 0.5 or 0.4. I care about one that's 0.00001. And a lot of those approximations, they don't work in the tails when all you're doing is focusing on mean and variance. Okay, so the Cauchy combination test is remarkable because the heavy tails of the Cauchy distribution actually ensure robustness to the correlation between the p-values. This was a very interesting result over here. And so essentially what it's saying is that if you use a heavy tail distribution, you can actually back transform over there, and at least in the tails, you don't really care about the correlation structure there. If you're far from the tails, it makes a difference. But if you're in that really lower tail, you're basically safe. So you can use other heavy tail distributions that people have used, the Levy distributions and some other families of distributions there as well. They work fairly similarly. So, okay, our modest contribution here is to notice that large p-values can reduce power. And if you have a p-value that is numerically exactly 1, then your corresponding test statistics is negative infinity, which means that your p-value is automatically 1. So very, very minor tweak on this. Basically, we find some point t, and if for any p-values that are greater than that t, we just set them equal to t. So the reason is if your p-value is 0.9 or 0.5, I don't care. I'm just going to set them all to be 0.5, and it ends up that we are totally fine in this case. If you futz with these really large p-values, it actually doesn't affect what's really going on in the tails. So we're usually okay. All right, so I'm not going to say too much more. The approach is fairly straightforward. We did some simulations, just 2 treatment groups. We simulated exponential survival with two different exponential parameters. All right, group 1 was simulated from clustered and group 2 from complete spatial randomness. We compared our proposed approach to something called SPF, which treats a summary statistic as a function of the radius. And so then they use functional regression models over here, which in principle sounds like a great idea. It doesn't really work well in the boundaries. And actually the approach doesn't work that well in terms of type 1 error. Their fun space is something that basically captures some sorts of entropy measures across 2 or more cell types. We also compared this to what if we just chose the best radius, or what if you just chose the worst radius within a particular application. And so here are our simulation results. Over here, we have the method, spot, SPF, fun space, and then choosing best or worst radius. And these are the type 1 error rates over here when we just have a single cell type. And so you can see that that for spot, and it seems approximately okay. SPF is slightly inflated, but not too bad. I think it's just the functional models don't work very well in these cases with these sample sizes. And the power, well, it is what it is, okay? Down here, choosing the best radius, as you would expect, your type 1 error rate is through the roof. It's it's not even worth talking about power in those cases. 2 cell types, similar story over here. But here we start seeing a little bit more difference. And in general, our approach tends to do better than the competitors. 1 cell type, not too much difference. There's slight inflation there. With 2 cell types, it ends up that we do quite a bit better. I think part of the reason is that when you're treating this as a function, you're assuming that there's some sort of, that the profile is somehow related, versus our approach assumes that there is a particular set radius or small set of radii where the association is very, very strong. So there's a slight conceptual difference and technical difference in that case. All right, so this just shows you the power across a different range of radii. And so you can see that depending on the radius that you choose, you do have very, very different power. Okay. We didn't apply this, we did apply it to 1616, but here's the analysis of a particular ovarian data set. They had 128 samples, and so there they looked at B cells, macrophages, CD4 cells, CD8s, and tumor cells, and we looked for associations between co-localization of cell types and overall survival, and we looked for across a range of radii, and these are the corresponding results over here. So you can see that at least for some of ours, there are some things that are recapitulated what they found in there. Your CD4 T cells are closely interacting with macrophages in these cases, and your macrophages and B cells are as well. Okay, what we have over here are the p-values for the association between overall survival and actually we use Besag's L in this case at each radius. And so the blue lines, the blue dots over here is looking at the interaction between B cells and macrophages, 2 different cell types. All right, and then the red dots are CD4 T cells. Radius is along the x-axis, the negative log 10 p-value along the y-axis. 0.05 is this dashed line over here. And so you can see, all right, if you choose a really lousy radius, all right, you're going to lose power. If you choose a good radius, all right, you end up with statistical significance. That's something to remark about. Secondly, the choice of radius is the best of correct radius or that you want, or ideal radius is different depending on the cell types that you're looking at over here. And that's just because, well, not all cells are created equal, and and not all cell types are created equal in this case. Okay, so we applied this to 1616 data. So we applied it over here, and we looked at a co-localization of individual cell types. Across all of the possible combinations, and we found that CD4 T cells are interacting heavily in tumor cells, and that is very prognostic or predictive in this case of response. And if you look at the on-treatment biopsy, all right, so this is after patients have started taking some drugs over here, then you can see that CD4 T cells and tumor cell interactions, as well as CD8 and tumor cell interactions, are again indicative of how long you're going to survive. Okay, so just a couple comments on this part, and then I'll move on. So SPOT, it's testing an association between spatial arrangements of cells and outcomes across different radii. It accommodates basically any spatial summary and outcome model that you really want, that you would typically use. Power seems reasonable. The type 1 error rate is pretty good, particularly compared to other approaches. All right, there are still things that need to be tweaked. You need to select an initial range of radii. All right, you can select one that's pretty wide if you don't know. If you have some ideas of what might be going on, such as based off of our prior analyses, then you can select something that's a little bit more restricted. The choice of summary statistic is also something that is ambiguous. You can also do an omnibus test across that too, using your quotient combinations. We're just too lazy to do it. Another challenge in spatial proteomics is how do you deal with multiple biopsies and slides at a single time point, and how to accommodate multiple images per person. And I am not going to talk about that. In fact, I'm going to skip all of that completely. And instead, I'm going to go to something which I think is more interesting, at least, which is how you do topological analysis of spatial proteomics imaging in this case. So what are we doing over here? So previously, we're talking about looking at cell arrangements. So we're just taking a cell and we're looking at their arrangements purely spatially, right? You're just drawing a circle around each one, and then you get a summary statistic based off of these. And so if you look across a bunch of them, then basically what you get is you get a bunch of curves over here across different samples, right? So again, you summarize spatial arrangements by quantifying radius R neighborhoods using some spatial summary, and then test for association between the summaries and the outcomes over here. All right. And so you can do this, select a single radius, you can evaluate multiple R, or you treat it as a curve in a functional model, or you can use the spot approach. So an alternative way to think about this is to look at the arrangements topologically. We're also doing work where we're doing things beyond this, but let's just start with something very, very simple, which let's just look at the arrangement topologically. So topological data analysis over here is really concerned with looking at the shapes of your data and shapes within your data over here. It assumes that the underlying data generating process has some shape in there, okay? And what you end up observing are shapes subject to some error over here, right? So then the goal typically in topological data analysis is to somehow recover or summarize the shape of the random process that is generating your data. And so here's an example, all right? You might have an underlying process, which is generating underlying shape, which is a circle or a ring over here. And then this is what you might observe in this case. So it's basically just a very noisy ring or noisy circle. And so that is the idea that we said, well, I wonder if we can use these within the context of spatial, within spatial proteomics. So visually, what we observe is this guy over here. This is a particular image from a breast cancer study. And then a hypothetical data generating process would be one in which, okay, you have rings so those are the red loops over here. And what you have over here are what we call connected components. So all of the points within here are in some sense or another somehow connected. Those are what we refer to as homologies types, okay? So red area, and that's just what I said, red and yellow areas are homologies. Homologies you can think of are basically degree K holes over here. So a degree 0 hole is just a connected component. The 2 points are connected. A degree K1 hole is a loop over here, okay? So it's a ring. So there's a hole in the middle. A degree 2 one is basically a void. So it's like a sphere with a hole in the middle over there, okay? And so this is one way of approaching this. And so the idea, the intuition here is that large homologies reflect the shape of data generating processes over here. And so the objective within a particular area called persistent homology is to identify very large or homologies that exist across a large range of scales over here. And this is done by constructing a filtration. So here I'm going to give you an example of how we construct a particular filtration called the Vittorius Rips over here. And so here's the original image. This is actually what you see over here. And we're just looking at this, in this case, at some tumor cells. And so it's an iterative process over here. So what happens is for a whole bunch of radii, you draw circles of radius R around each cell over here, each point in your data. Then when circles touch, all right, they join together and become a single group over here, all right? You draw an edge. And so what we do is we record the births and the deaths over here of connected components, all right? So 2 things coming together, as well as loops over here. So rings that suddenly form in your data. And the idea here is you want to observe homologies, so loops, rings, loops, as well as connections that persist, right? So things that are there across a large number of scales, which suggests importance over here. So here's an example. So what you start off with is each point is basically itself. And then what you do is we draw a radius 50 around it. And so then you see these little purple circles. That's just radius 50. And so you can see already at 50, some of these guys are touching. And so you say this is basically a birth of a connection over here. All right. And so on and so forth over here. Okay. And so you can take a look. That's what's happening. All right. So there's now some connected components. These guys are connected. This guy over here is already connected to a whole bunch of other things. And you move from there. Let's increase the radius at 100. All right, these are the different things that you see. So you can see that this guy is still there. It still exists across there, but these guys are now connected. So the individual points have died, but then now they form a new connection in this case. All right. And if we keep increasing this, eventually you start seeing rings in your data as well, right? And so you start seeing some loops that appear. And if we keep going, well, those loops have now disappeared, right? They've filled in, but you have got new things that are starting to form in this case. This one still persists versus some of the others do not. And so if you go to a much larger radius, then you see now that what you have over here is some degree of structure among your cell types over here, among this particular, among your cells. So the idea behind this is that you're really looking for structures in your data. So it's more of a physiologic phenotype in this case, right? It's not just whether cells are close to each other. It's still related to spatial because of the choice of filtration that we used. But the idea is that you're looking more globally at this and you're looking for larger scale structures, which could be within the context of cancer, reflective of vascular tissue being like blood cells and things like that. Not blood cells, blood vessels and things like that. If you have a lot of blood vessels, your tumor is getting lots of nutrients and maybe it's growing bigger because of that. All right. So again, so what we typically do then is you take this original image, and then for each image, you generate something called a persistence diagram over here. And so what you're doing with these is you're looking at the births of connected components or loops, all right, across different radii over here, basically, across different times. And so what you have over here, these are your connected components, and then these are your loops. So they're completely different. We separate them out in this case. And then the lifespan of each point is basically its distance from over here. Like how long, what is the lifespan of this thing? And so you think, all right, this guy persisted for a long time. That suggests that it is perhaps more interesting or more important somehow. So for each person, you have their original image. You put it through this filtration, and from there you generate a unique persistence diagram. This is now unique for each patient. So each person has one of these guys in this case. Okay, that's great. Now what do we do with it? So that's where people don't do much statistics over here. There's been some work over here, but not a huge amount of it. So remember, I don't actually care about that much about the topology of the thing. I care about how the topology relates to an outcome. All right, that's what makes me interested in biostatistics. I care about people and whether people live or die. So the question is, how do we assess whether the topology is associated with clinical outcomes? All right, well, it ends up, as I said, each patient has their own persistence diagram, which is a representation of the image's topology based on births and deaths of components and of connected components and wholes in this case. All right, so our proposed approach in this case is very simple. All right, we're going to assess whether the persistence diagrams are associated with outcomes. Persistence diagram in this case is basically one-to-one with your topologies in some sense. And so the idea behind this is pretty straightforward. And so this is our method over here. The name is still under negotiation. I call it the Persistent Homology Analysis Regression Test, PHART, or if you combine PH, makes an F sound. All right, it's the FART method. My postdoc is a bit of a stick in the mud, and so she doesn't really like that name because she doesn't want to go on the job market and have to present a method called FART. But I think it's awesome. But I also developed a method called SCAT. So I mean, which is another word for poop for those of you who are not native English speakers. Yeah, but again, I have a pretty immature sense of humor. So I'm going to call it FART for now, but other people, the name may be something else eventually. Okay, so here's the FART approach in general. So the idea here is you take your topological information, you summarize it as a persistence diagram. You can actually measure distances between persistence diagrams. There are standard metrics that people in math and other areas have developed. And so a distance, all right, how different things are, you can flip it around basically to become a similarity, all right, which is what we call a kernel. And then once you have a kernel, you can use a whole area of biostatistics called kernel machine testing in order to do the association analysis. So here are the specific steps. We apply a filtration to each image to generate the persistence diagrams, all right? Then we use standard distance metrics to calculate distances between persistence diagrams based on each homology group. So these distances are only used for loops, or they're only used for connected components, or if you had higher dimensions, they're only used for rings at any given time, right? So you get a set of distances based on components, a set of distances based on the holes or the rings in this case. Then we convert distances to similarity. So distances are a dissimilarity. Now we basically flip it around to become similarity, which we call a kernel. And then we can apply standard kernel machine regression testing framework. All right, so how do you compute distances between persistence diagrams? There's bottleneck distance, which is looking for this largest single difference between corresponding points on two persistence diagrams. Basically, you order them, and then you see sort of what is the maximum difference over here. Okay. Wasserstein distance, again, another commonly used distance. We use actually in many different areas of biostatistics. It's basically the minimum total difference between all matched points over here. Okay. Then there's total persistence, which is basically some sort of L1 distance between the ordered lifespans in this case, right? This guy over here is technically not a distance. It does not satisfy certain triangle inequalities, but it's still a dissimilarity, and we end up using this for the most part, mainly because these guys are computationally really slow, and you know, I want to write a paper within the next year, not the next 10 years. So it's totally practical. These guys are probably better. Theoretically, they're better. In practice, they're probably better. But again, I'm a very pragmatic person. This is good enough. All right. And there's many others that people used. And so from this, you can get a distance. Now, we have our distances. There's a distance between you and me, okay? Between me and every other person, and between you and every other person in this room. So you end up with a big matrix of distances, of pairwise distances between samples, okay? Between people in this case. There's several strategies for converting distances to kernels. Within the kernel machine testing literature, what we do is basically do some Gower centering, and then that inherently flips it to become a similarity over here, all right? The approach that's used in the topological data analysis literature is that actually they exponentiate this using some sort of Gaussian, where they take a Gaussian kernel and then they're just replacing the usual squared distances there with this other distance over here, all right? Again, for the most part, we're using this guy over here, but I'm not sure, and we have not played around too much with using this strategy. Okay, as I said, there are distinctions between distances and dissimilarity. If you use a distance, then this guy over here is guaranteed to be positive semi-definite. If it's a dissimilarity, you no longer have that guarantee. We found that actually, even with negative eigenvalues, it doesn't really make much difference. But if you want, you take all of those negative eigenvalues and you set them equal to 0, and you know it's sort of a loosey-goosey way of dealing with it. Again, very pragmatic. So, all right, the last step of FART, p-HART, or all right, is kernel machine testing. So again, all right, we're now at the point where we have the kernel. The objective is to assess the association between topology and outcome, such as survival, all right, which is, well, a censored survival outcome, change in tumor volume, which is a continuous outcome, or response, which is a binary outcome, right? So you have all sorts of different outcomes that you want to deal with. So for a given homology, okay, a general test statistic, and there's many different derivations of this, is basically this guy over here, where R's are some sorts of residuals from a null model between the outcome and the covariates. So within the context of linear regression, these are just some sort of standard residuals. So this is your outcome over here. So change in tumor volume, these are covariates, age, sex, whatever you want to stick in there. And then R's are just the residuals over here. Okay. If this is survival, then this can be your Martingale residuals. If this is from a GLM, then it's basically looking at the linear model at convergence of your set of linear equations over there. Okay. So you have some notion of residual, and then this guy over here is basically looking at the correlation between the residuals and your kernel. That's all it is. Nothing terribly fancy going on here whatsoever. And so we have done this ad nauseum in the genetic association analysis literature. And so this is a very, very standard approach. Nothing fancy. The null distribution of this guy, it's actually a mixture of chi-squares. There's lots of finite sample approximations for small samples. which basically involve calculating the moments of the finite sample permutation distribution. And so you can end up with some nice finite sample results, which are kind of messy, but remarkably useful. If you have, so in our case, we can get a p-value if you have just a single test statistic. In our case, you have two test statistics, one for order 0 and one for order 1 homologies over here. And so what we do is we basically calculate a weighted sum of these guys, and we obtain a p-value for each weight. Well, now you have a p-value for each weight. How can we combine those? Cauchy combination test. Nothing fancy overall. So this is not a terribly fancy approach. We're not doing anything statistically that innovative, but we're just putting some things together. Okay, but it works. So here are some simulations to show it. We compared persistence, our approach to basically standard spatial analyses. And so here, it's again, we're looking at survival outcomes because that's really what we're more interested in. We looked at powered type 1 error rates. So these are the simulation results. So these are the topological analysis approaches, looking only at the order 0, only order 1, and the omnibus testing. These are spatial approaches. And so this is a situation where we generated group 1 to be from one type of structure versus, and group 2 was complete spatial randomness. So they're squares. And so you can see the power and the type 1 error over here. Then what you have over here are loops versus complete spatial randomness, loops versus squares, and then clustered versus complete spatial randomness. And in this case, all right, overall, looking at this with a persistent homology approach tends to do pretty well across all scenarios over here. And our type 1 error rate actually seems a little bit better as well than some of the other approaches. But again, you know, it is what it is over here. But basically, it seems to do reasonably well, and where there are differences, ours tends to win. Yeah. Okay, so I'm very low on time, so I'm just going to go to a data analysis over here. So this is a MIBI study of 38, so it's a very small sample size of 38 triple-negative breast cancer tumors. And so then the spatial analysis actually observed three different categories of tumor microenvironment. And this was done using a very, very fancy approach called eyeballing the slide. And so they looked at it and they're like, hey, there are three different types. These guys, everything is mixed up. The immune cells and the tumors are mixed together. This one, they are totally in separate areas. And this one is a cold tumor where there are basically no immune cells. So they're not infiltrating in there. They're not attacking the tumor whatsoever over here. And so we said, can we describe these categories topologically? So we applied the persistent homology analysis, FART to immune cells. I can't even remember what it stands for. I don't care. I just like the acronym. All right. And so what we found, for example, is that mixed and segregated TMEs showed very significant topological differences over here. So this is the mixed. You can see that everything is sort of down here versus once they are segregated, you start seeing more structures in the data in this case. All right. And so the p-value in this case is 0.00005, which is a very happy p-value for us. And so you can see over here that there are some pretty significant differences. And these are just example images, right? And then one of the nice things is we look across the different range of radii, and to try and interpret what's going on, we looked at the best RIPS radius where we saw the most separability. And so this is what you're seeing with a compartmentalized TME, so tumor microenvironment. And then this is an example of the mixed. So you can see that there's a lot of connections here, but then over here, you're really starting to see much more serious and well-defined loops with fewer of those sort of just these very sparse things going on over here. Okay. So just to comment, this is just the beginning, all right? And we think there's a lot more work to do in this, all right? And I'm going to skip over this discussion, not that exciting. Instead, I'm going to talk a little bit about what I'm excited about, which is actually looking at something that we call cellular orientation. All right, not just, all right, so cells, all right, they're like people in a room. They're just all sort of standing around. But the thing with people in a room is you're facing a particular direction. You're facing this way, and you're perhaps walking this way, facing this way, walking this way, all right? And so we can actually get some idea of the mechanical directions that cells are growing and dividing using spatial proteomics. And so this is what you get over here. These little arrows are basically the directions that cells are facing. And within here, you can start seeing very, very interesting structures. Here's a source. Everything is going away from there. You have certain sinks where everything is coming in. You have curls, you have divergence. You have all sorts of interesting things that you might get from a vector field. I looked through the literature. Nobody has developed statistical approaches for vector field analysis. So I'm excited to see what we can start doing with some of these things over here. Lots of opportunities. We can calculate curls, we can calculate divergences, lots of strange and wonderful things that I'm excited to pursue. Supposed to give this talk in December. I was hoping to have more results by then, but it's like, Halloween works too. Just some acknowledgements. Sarah did most of the heavy lifting over here. People from the translational study team. A personal plug. I'm always looking for more people to come join us. All right, we have a great group. Taylor doesn't know that she's a part of our group yet, but she's honorary over there. Okay, and we also do other fun things as well. I'll take any questions in the next 2 minutes before people have to go. All right. Yeah. I know. I put on clothes today. Like, seriously. I actually had a student tell me that when I was lecturing because I broke a strap on my sandal. And so I started lecturing in introductory probability barefoot. And so another student came in late and I heard her say to the other one, he's not wearing shoes. And she was like, just happy he put on pants. Yeah. Yep. Yeah. So they're actually, you can look at the proteins within there. And based on the angles, based on how they are forming and they're orienting, you actually know sort of which way they're sort of dividing. So they are estimated from the data. Yes. Yes. Uh-oh. Yes. Yes. I don't think so because no, the reason is that these are not our analysis units at the end of the day. The analysis unit is the person. So everything that you're talking about is measurement error, right? In our case. And so you're right. I'm not taking into account any degree of that measurement error, but I don't think it's strictly speaking double dipping. I think my analyses are valid from a type 1 error standpoint, looking at phenotype. Yes. Yes. Yeah, with all you're doing is analyzing a single image to death, then I think you're in trouble. But that's, yeah, I always struggle with that. And I have to admit, I actually don't even like those spatial transcriptomics things because I don't even understand the sampling unit over here. Anytime we do frequentist statistics, right, we're always talking about a sampling unit for randomness. And any analysis that you do with just a single image means, hey, I'm going to take more cells from the same person. Well, you've already imaged their whole brain slice. So I don't even understand what that means in this case. So anyway, sorry, I'm ranting about things, but in my case, I get away from that. So yeah. 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 Again, once you get to the population scale where it's person, it's okay. I mean, I do have measurement error, absolutely. And that could affect things, but I'm just sort of hopeful that, it's additive in some sense or whatever, and I can just sort of ignore it. So, yeah. Yeah. Yep. Yeah. So typically, I mean, histology is one thing, but typically for a lot of these, right, the stage and a lot of those things, we try to adjust for them within the regression model, all right, where necessary. Oftentimes for stage, because there's such big differences, we oftentimes just stratify the analyses. Histology, probably similarly as well. We're not explicitly taking that into account, and for the most part, if you have, say, luminals versus basals and things like that, we sort of just ignore that, at least in these analyses. That could be an endpoint, or it could be that you're just or it could be that you're just going to have to focus on a particular histology in this case. As for where it is coming from, that is a real challenge. Because what we found, for example, is, well, here you have your giant melanoma over here in some cases. And so you poke this place and get a biopsy versus you poke this place. They actually look totally different. And that is a real problem. And so what we're trying to figure out are what are ways that we can actually combine images taken from the same patient at the same time point. Within spatial summary statistics, there's all sorts of averages that people have developed, and those seem to actually work reasonably well. We have a paper in Briefings in Bioinformatics. It's in press. I think it's come out already, where we explore some of those types of things. But it is an excellent question. The best way to do it, I've not seen. Some people try to use some sort of mixed models to deal with these sort of repeated measures and things like that, but I don't think it works out that well. The type 1 error rate, at least that we've seen in practice, is pretty awful. But good question. I don't know the answer to how to deal with the fact that there is heterogeneity. If it's a known variable such as histology or stage, then that is something that you either stratify or adjust out. Yeah. Yep. Yeah. Yes, it's always from the primary in this case. Yep. We didn't look at any of the Mets in this cases. Actually, we may have a few Mets, but they're not in this analysis at all. Yeah, this is actually a very interesting data set, and that should appear probably, well, hopefully in the next few months, also in a nice journal. So, yeah. But all right.
Yeah, and thanks so much, Mike, for a very
Yep.
interesting talk.
All right. Thank you.